Thank you. Good morning. I'm really super excited to be here today for a few reasons. The first one, it's like many of us, my first international traveling for 18 months. Second, I'm back in California, which I really love. And the third, and not last, or not least, is that we've announced today a strategic collaboration with Intelia, and I'm going to speak about this transaction in the course of my presentation. If I can get the presentation, and apparently I cannot. Well, I will do without. That's the challenge of being live. So SparingVision is a company that was started in 2016 as a spin-off from the Institut La Vision in Paris. We were started with the idea of disrupting the field of genomic medicine in ophthalmology. Our legacy asset, the asset that, on which the company was started, is called SPVN-06. It's a gene or mutation agnostic gene therapy that we are developing in RP. The way this product works, unlike gene replacement or gene augmentation therapy, is that we have in an AAV a cargo, which is a neurotrophic factor combined with an antioxidant enzyme that will act synergistically at preserving the cone in rod cone dystrophies. So in the example of retinitis pigmentosa, but this is a similar pattern in all these kind of disease. You have a slowly progressive disease, turning you inevitably, when you are affected with the disease, into legal blindness. And the reason why we believe this is the case, despite the fact that it is all starting always starting with the degeneration of rods. At one point, what is critical for the vision, which are the cones, will in turn die, despite the fact that they are usually not affected by the mutation that has caused the disease to start with. And what the scientific founders of the company have found and discovered and deciphered the mechanism of action of over time is that the rods are responsible for the supply of the cones with a critical neuroprotective factor that we call RDCVF, or RON-derived cone viability factor, that is, again, acting on the metabolism of the cones so as to preserve them from dying or further degenerating. So the idea of SPVN-06, our lead asset, is to come at a time when most patients are diagnosed, so it's not early on, it's when, unfortunately for them, they have lost peripheral vision, they have lost some night vision. They go to their physician, and independently of their genotype, because they have not been genotyped at that stage, they are diagnosed with RP. And at that time, they are no longer eligible for gene replacement therapy or gene augmentation therapies, which are going to, again, target mostly the rods. They are left with only cones, and SPVN-06 will be injected at that time so as to preserve what is left of the vision at the time of diagnostic, at the time of intervention with the product. So what we are doing basically with SPVN-06 is that we are putting the neurotrophic factor on the enzyme that is a very, again, potent antioxidant enzyme. Here are the slides. Okay, thank you, guys. I'm going to, try to go back in time. And SPVN-06 is the lead asset of the main pillar of the three-pillar portfolio that we are building. And this asset is going to, if everything goes well, enter into human testing, sometimes in 2022. Next to it, we are building a full set or a full suite of a product along the same line, i.e. mutation-agnostic gene therapy product, SPVN-06, SPVN-20, which, unlike 06, which is going to work on metabolism of the cone, SPVN-20 will act on the function of the cone, and the idea will be to restore function in what is called dormant cones, which are cones which have lost their ability to function as part of the evolution of the disease. And then SPVN-30, the last product of that bucket or pillar in our portfolio, is a product that will combine the transgene from the first two products. All these products are AAV, and all these products, at least for the first generation, will be injected subretinally. News of the day. This morning we've announced a collaboration with Intelia, and that collaboration will be filling the second pillar, the second bucket of our portfolio, which will be made of genome editing product. And as you can imagine, we are super excited to have been selected by Intelia to explore their CRISPR toolkit in the space of ophthalmology. And then, but this is probably a little more futuristic, we are building a third pillar in vivo reprogramming. The idea being in this pillar to find or identify transcription factor that we will put into a viral or non-viral vector and try to turn some cells which are not critical for the vision into cells which are critical for the vision. As you can guess, if we manage to do that, that will solve many of the blinding disease problem. That's the portfolio. So yesterday evening, we had only 3 products in the portfolio from the first pillar. After the announcement this morning, we are adding 3 products. These are potential products, obviously. We need to select the target and move on. But as you can tell from this slide, despite the fact that this is early stage, this is already a totally unprecedented portfolio in our space. I'm going to skip this one in the interest of time. RP, which is the disease on which we are going to focus the development of the gene therapy franchise, is following a pattern which is common to all rod-cone dystrophies, which is illustrated by the painting that you can see on the bottom of the slide. It's a painting by Renoir. It's called Missy-Sanrio. If you want to buy it, you will have to spend a lot of money. But the point here is today is that on the left side of that painting or that slide, bottom left side, you have the painting as you and me, if you are not affected by RP, can see. And then progressively over time, unfortunately, you will start to be tunneled into darkness, and eventually Missy-Sanrio will disappear. We categorize the stage of the evolution of the disease into 4 different stages, and the idea of what we are doing will be to intervene with 06, 20, and possibly 30 in stage 2 and 3 of the disease, where you find today 80% or so of the patient population. Again, 06 will act on the metabolism of the cone, 20 will act on the function of the cone, and specifically will try to reactivate the phototransduction cascade in dormant cones, which are viable cones, which have lost the the ability to function as part of the evolution of the disease. And then 30, again, is a combination of the 2 products. I'm going to skip this slide in the interest of time. SPVN-06 is an AAV product, and as you can tell from this sketch, we are putting into a single AAV 2 transgenes, which are 2 isoforms of the gene NXNL1 that was identified by our scientific founders more than 10 years ago, and we are attaching those different transgenes to 2 specific promoters. And the idea, again, would be to inject the product at a time when rods are gone. And we would like RDCVF, which is the neurotrophic factor normally expressed by the rods, to be expressed in other cells, since the rods will be gone by that time. And so we are attaching that transgene to an ubiquitous promoter that should lead to a broad expression in the tissue or environment of the cone. And then RDCVFL, which is the antioxidant enzyme, which is the second isoform or the other isoform of the same gene, will be attached to a cone-specific promoter so that it is expressed specifically in the cone. I'm going to go very fast on the preclinical package, but what I can tell you is that we are seeing very nice activity in the morphology of the retina. What you can see on the left side of that slide is a retina of the pigs, a transgenic pig, and as you can tell, if you were to spend a lot of time on it, when you inject the product and you compare that product injected to an eye that has received a control, there is a clear difference in impact in terms of the morphology of the cone. And then in terms of pharmacology activity as well, we've been looking at function or functional readout in mice model, and not only we see the kind of morphology impact that we see in pigs, but also a very clear-cut impact on the function of the cones as measured by visual acuity. Those data are not published, and I will not show them to you today. On the right side of the slide, this is a summary of a pilot tox study that we've conducted in NHP earlier this year, and which is showing a good safety profile. Where we are with that product, and I'm going to go very fast, we expect to be IND ready by next year. So we have completed the pharmacology package. We have a GMP batch ready to go. And the last thing on the critical path to IND and CTA submission is a GLP NHP tox, which is starting in 2 weeks' time. Rapidly, SPVN20 mechanism of action. So it's a different transgene. Here we are no longer looking at preserving the cones, but restoring some functions. So we are going to introduce a transgene which is called GIRC, which effect we believe will be the restoration of a short phototransition cascade. Very rapidly, some nice data to show that both in terms of photopic response as well as visual acuity, we've been able to see a clear and statistically significant difference between eyes receiving the product versus control. This product should have its lead candidate selected in 2022 and then should go into IND and CTA submission in 2024. This is the positioning of the gene therapy franchise. We are right in the middle, again, where the bulk of the population is, unfortunately. On the left side, you will find all the gene replacement, gene augmentation therapy, which for most of them are mutation-specific. On the right side of that landscape, you will find optogenetics and transplantation, which, based on what we are seeing today, we believe will be, at best, restoring some part or some fraction of the vision. So the unmet need for what we are currently doing is just enormous, unfortunately. Genome editing on the news of the day. So we've announced, as I was saying earlier today, a strategic alliance with Intelia. We believe it's a very good complement to our first pillar of mutation-agnostic gene therapy product. Needless to say today that Intelia was possibly the best, or surely the best possible partner for that franchise, given their expertise in the field of genome editing. I'm sure you've all seen the data that they've released earlier this year in the liver, and we went to see them a few months ago and tell them, look, we are building a world leader in ophthalmology and genomic medicine. You have a fantastic toolkit. You are not doing anything in ophthalmology. Let us become your partner in ophthalmology, and we share the economic benefits. And that pitch has kicked in and has led us to the deal that we've announced this morning. Since we are a Series A company, we had no cash to pay for an upfront, and so very early on we told and convinced Intelia to receive our shares in contribution of the upfront, and they have accepted, and so they will own 10% of SparingVision. Then they will be eligible to cash milestone, and if everything goes well, royalties on sales. They will have opt-in right so that they can get the US commercial rights back at one point in the development of the compounds. And of course, for that, they will have to pay an upfront to us in cash. They will have to pay or reimburse some cost, and then moving forward, we would share the cost of bringing those products to the market 50-50. I'm going to skip this one. Needless to tell you or to speak about the potential of CRISPR in the ocular space. What I can tell you is that the first human data were released publicly by Editas a few days ago, and we know that they have been, let's say, criticized. I personally believe that the critics were unfair. We all have to always remember, always bear in mind that phase 1 is mostly about safety, and what we've seen of the safety profile of that product is very good, very encouraging for the field, for Editas, for the patient, and for us. We probably can do better when it comes to efficacy, and we will do better when it comes to efficacy. But what is good for us right now is that we know that when you put CRISPR into an AAV and you inject that product via the subretinal route, it's safe. And that is a very good starting point. And we hope that for Editas, for the field, and then for us, efficacy will come later. In the agreement with Intelia, we have a basket of predetermined targets that SparingVision will have the sole discretion to select in the years to come. And this basket of targets is the result of a very thorough analysis of the field and of the 250 plus ocular targets that are known today. And we've been selecting that basket based on probability of technical success using CRISPR to address a particular target, the unmet medical need, obviously, and the prevalence of the underlying disease, the competition, and last but not least, whether that a particular target was addressable successfully, we believe, or with a high likelihood of success, with gene replacement or gene augmentation therapy, because in that case, it would probably be pointless to go with a more, let's say, technically advanced product. And so we have in the contract that list of predetermined targets, and in the years to come, we are going to select one by one up to 3 targets, and I hope that we will be selecting the first one in the weeks to come for a development that could start in 2022. My last slide for today, and thanks God I have the slide. In short, SparingVision, we are on our way to really disrupt the field of genomic medicine in the ocular space. Our lead product, we believe, will be the first mutation-agnostic gene therapy to enter into human testing as soon as next year. We have some new pharmacology data that were gathered during 2021 that is leading us to lower the dose. And in gene therapy these days, if you can go lower in dose, it's probably better, because gene therapy are under scrutiny with the regulators for some very, very legitimate reasons. So we've managed to identify a therapeutic window that will be at a lower dose than what we initially were anticipating. And that's good. We are going to use that new therapeutic window in the upcoming GLP NHP tox study, which is starting again in the days to come. And we expect IND and CTS submission in the third quarter of 2022. SPVN-20, our next product in line, is on track for an IND and CTS submission in 2024. We are going to select the lead candidate mid next year, and then we'll start GMP development in 2023. We can expect multiple, catalysts from the partnership with Intelia in the years to come, and we are going to continue to expand the pipeline. Probably the next pipeline expansion momentum will be in the in vivo reprogramming bucket that I was referring to before. And we are still a small company, 20 plus employees, Series A, 60 million euro raised so far. But thanks to the maturation of our pipeline, thanks to the Intelia collaboration, we are going to aggressively expand, both in terms of headcount, in terms of infrastructure, and in terms of fundraising. With that, I'm on time to finish my presentation. Thank you very much for your attention today. Bye-bye.